Hi, welcome to our BioBits Central Dogma Lab webinar. I am Dr. Katy Martin. I'm going to be demonstrating this one-of-a-kind experiment for you today. The BioBits Central Dogma Lab is one of our most popular learning labs. It's a hands-on way to explore the central dogma, a curriculum area that's usually hard to supplement with scientific experimentation. We're reintroducing this unique lab to you today because we've recently updated the curriculum. We've expanded it, we've written more of it. We've also broken it up into modules that you can kind of mix and match to tailor this lab better to your students, no matter who they are, whether they're in middle school, high school, or college and beyond. We've also added new digital tools intended for use with this lab that give you a bit more bang for your buck. They give you more flexibility around how you implement this lab, and they also support greater student engagement. So if this is a lab that's already familiar to you, you should learn about some new curriculum today and new tools that you can use to make this lab even more kind of customized to your group. If you're new to the Central Dogma Lab and you're not already familiar with it, know that this is a lab for every biology classroom. It's a great way to support hands-on instruction around this essential biology topic. In our session today, I'm going to begin by just giving you an overview of the Central Dogma Lab. We're going to break and do some hands-on work. I'm going to set up my samples so you'll see how the protocol looks when it's executed in real time. There'll be a brief incubation, during which time I will kind of back up and explain the samples that I prepared. We'll talk through predicting our results. And then I'm going to close by talking about classroom implementation, explaining what it would take to bring this to your students and the different options you have for customizing it and differentiating it. Before I even dive into that, though, I just want to back up and introduce Mini PCR Bio to those of you who aren't already familiar with us. We're a company that cares about making science accessible. We do that by making lab equipment that is different, that can go places that traditional biology tools cannot go. So we have scientists who bring our tools into the field to do DNA analysis on site where they're collecting their samples, whether that's in the jungles of the Amazon or on a research vessel traveling the Arctic. We have users who bring our tools into their own homes to pursue their passion projects. So this is a self-taught mycologist. He uses our equipment to identify the mushrooms that he forages for and that he cultivates. But most of our tools live in classrooms. They're used by teachers and students. And so we make classroom-friendly tools. We also have a curriculum team that writes labs so students can wrap their heads around the types of problems that can be solved with biotechnology. I'm a member of our curriculum team. I am down here in the lower left-hand corner holding an alligator. And my background is similar to those of my colleagues. I trained as a scientist, so I studied neuroscience in grad school. But I felt like my home was in the classroom. So I taught high school for a few years after graduating, and I've been on the Mini PCR Bio team now for about 6 years. My current focus here is on heading up Mini PCR Digital, our e-learning platform. The perspective that my colleagues and I bring to writing curriculum is that biotechnology activities, hands-on lab activities, they can make fundamental biology concepts come to life and really enliven students. So biotech to us isn't like a 1-week unit that you slot in at the end of the school year after you get through the important stuff. It's actually a lens through which you can cover that important stuff in the first place. And the Central Dogma Lab is a great example of that. In this lab, students will watch as RNA and protein are made in real time. So they will watch the processes of the Central Dogma Lab as they take place. They'll also learn how gene expression can be disrupted. This lab comes with a free curriculum. You can go to our website and download it anytime, teacher's guide and student's guide. But we've also added a digital lab that can be used either with or without the hands-on kit. And this virtual lab offers really great visuals to students to support better understanding of the processes at play in the Central Dogma and how they interact with one another. I'm going to be using the visuals from the digital lab as we go along to illustrate some of the science that's happening, just to give you a sample of this curriculum option. So the central dogma of molecular biology, one of the most important concepts in biology, the foundation really upon which all of modern biology rests. And to those of us who love biology, this theory is a beautiful theory that explains so much about what we see across, all living things. But For students who are first becoming familiar with this content, the central dogma can be a really confusing topic and a really boring topic. So this is a textbook page that covers the central dogma. That content doesn't exactly leap off the page, right? So in the classroom, we supplement this with other activities to kind of help students imagine the processes of the central dogma. So virtual simulations are one option. There's a really great free virtual tool from Utah Genetics that simulates transcription and translation with beautiful visuals that really show students what the molecules look like, how they interact with one another. And this can really help enhance students' understanding of the central dogma. You can also use hands-on activities, so paper modeling approaches. When I was a teacher, we built a paper DNA molecule that stretched all the way across the classroom. And this really is fun for students, and it also helps them picture the molecules and the processes they coordinate in a bit more detail. But in a science classroom, the best thing would be to use science practices to investigate the central dogma. But why isn't there already a best-selling lab on transcription and translation? Well, these are processes that happen in living cells, and keeping cells alive is expensive and it's time consuming. So for teachers who are often working with very limited time and limited budgets, bacterial culture is just a bridge too far. It's too much of an investment to pay off. So in writing this lab, we were wondering, is there a way we can experiment with transcription and translation without this requirement of keeping cells alive? And the answer for us came in the form of cell-free technology. Cell-free technology has been used in biology research labs for decades now. The basic principle is this. You start with bacteria. You basically put them in a blender and harvest their guts. We'll call that cell extract. It contains things like RNA polymerase, ribosomes, tRNAs. We add to that cell extract supplements, building blocks like nucleotides and amino acids, an energy source like ATP, and together, those components make up the machinery and materials that cells use to build proteins. You can take this, put it in a tube, freeze dry it into a pellet. And if you've done that, then you've produced what we call a BioBits pellet. So BioBits is the type of cell-free technology that we're going to be using in this lab today. BioBits was developed by my colleague on our curriculum team, Dr. Ali Huang. She was a PhD student at MIT and she was studying cell-free technology. And she became interested in the idea of making that science, which is amazing, cutting edge, used in science labs. She wanted to expand access to that technology to students to aid in biology learning. So she was really responsible for developing the Central Dogma Lab. In our lab today, we are going to be adding DNA to BioBits pellets. DNA, of course, includes the instructions cells use to build proteins. When we do that, all of the machinery in those BioBits pellets is going to come to life. It's going to transcribe that DNA into RNA and translate that RNA into protein. And so basically, BioBits gives us a way to produce proteins to trigger gene expression on demand, according to the instructions that we provide the system. In this lab, we'll be using that BioBits technology to observe transcription and translation as they're happening in vitro, and then we'll explore different ways of interfering with those processes. We're going to be able to watch along because we'll be using fluorescence to track gene expression. Fluorescence is something that you encounter in your daily life all the time. So if you have a driver's license that anyone has ever shown a UV light on to authenticate it, that's fluorescence. It's when a molecule absorbs light of one wavelength and then spits it out at another wavelength. Fluorescence is used in the biology lab all the time. So one example is fluorescence microscopy. This is one image of some cells that have been labeled with fluorescent markers. So here, the nuclei of the cells has been stained with a blue fluorescent marker, and then we've used a green fluorescent tag to mark a cytoskeletal protein. So we're following in this tradition today in our BioBits Central Dogma experiment. In this lab, we are going to be producing green fluorescent RNA and red fluorescent protein. And so green and red fluorescence become visual indicators that transcription or translation, respectively, have happened. We're going to be able to watch along as that happens using this device, the P51 molecular fluorescence viewer. This is an affordable and durable piece of hardware that MiniPCR Bio makes, and it's a way that you can detect fluorescence from biomolecules like DNA, RNA, and proteins in samples in a simple visual way. So at this point, I've kind of given you an overview of our Central Dogma Lab experiment. I want to break and now do some hands-on work. I'm going to set up my samples so you can see what it looks like when this protocol is happening in real time. Then there'll be a brief incubation. During that time, I'm going to back up. I'm going to provide a lot more context and explanation of what exactly is in the samples that I'm setting up today. So here is my Central Dogma Lab kit. We're going to start with this. Inside there is where you'll find the BioBits pellets. So this is my strip of BioBits pellets. At the bottom of each of these tubes, you might be able to see something that looks like a grain of sea salt. That is our BioBits pellet. So I'm going to be adding reagents to each of those BioBits pellets using a micropipette. Today I'm going to be using this mini pet. It is a fixed volume micropipette, and it always pipettes 4 microliters. Now I'm using this mini pet today because it's a great classroom tool. Mechanically, it's very similar to a standard variable volume micropipette. So the one that looks like this, that has a little window there, you can dial in your volume. Mechanically, it's very similar to that variable micropipette. We have a plunger here on top that we use to aspirate liquid. There is a little ejector button here on the side that we can use to eject the tip when we're done with it. And then I'm just using standard micropipette tips here, not like special mini pet tips or anything. Just regular ones work fine. So mechanically, what students learn on this mini pet is going to translate when they pick up a variable micropipette. But because they don't have to dial in the volume, it removes one potential source of error. So it's a bit like pipette training wheels, this mini pet is. And I'm changing my tips here with every addition because I don't want to cross-contaminate anything. So that's important for this lab. Now, as I mentioned, we're pipetting 4 microliter volumes here. That's a pretty small volume and very easy for students who are first learning how to pipette to make mistakes pipetting volumes that small. So I definitely recommend pipette practice. A full day of pipette practice is great. 20 minutes is just fine. Something to get them comfortable pipetting before you turn them loose on the lab. It'll pay off in the form of better results. They'll get better results. They'll be more similar to the expected results. They'll be easier to interpret. So definitely recommend investing in some pipette practice before undertaking this lab. And even for those who are comfortable pipetting, there's a few things to know about pipetting into BioBits pellets that I want to call out here. So as I mentioned, BioBits pellets, they're freeze dried. And so they are super hydrophilic. When they come into contact with moisture, they want to go toward it. So if I take my pipette tip here when I'm adding reagents and I touch it directly to the BioBits pellet, the pellet is going to try to like jump up into my pipette tip. I don't want that to happen. So I'm just pipetting onto the side wall of each tube. And then I'm letting the droplet kind of roll down and hydrate the pellet. And I'll show you what that looks like here on my next tube. So I've got 1 tube left to set up, tube number 4. I'm going to try to do it so you can see what's happening. So this tube is the one I'm pipetting into. I'm taking my pipette tip, I'm just having it kind of sit next to the side wall of the tube. I'm pipetting the droplet onto the side wall and letting that droplet roll down onto the pellet. That's the technique you want to use when you're pipetting into BioBits pellets anytime. Okay, almost done setting these up. I do want to note that the protocol for this Central Dogma Lab is mini pet friendly. So it has been written so that you only need to pipette volumes that are multiples of 4 microliters. So you can use this mini pet to complete this lab. Okay, got all my samples set up now. I'm going to put the caps back on my tubes. And I'm going to take 2 quick observations before we start our incubation. So first, I'm just going to visually check my tubes and see that the volumes are pretty much similar across all my 4 tubes. They look pretty consistent to me. If you had a student group and you saw that their tubes had really varying volumes, that would be some indication that maybe they missed a step, maybe they pipetted incorrectly. If I had extra reagents, I would give them a new set and have them try again. Mine look pretty good though, so we're going to proceed to observation. So this is my P51. I am going to put my tubes right in the center wells of the P51. I'm going to turn on the light. The light switch is in the back of mine. And we'll just take a quick look at these before any time has elapsed. So yes, you can see my tubes. The light is on, but we don't see any fluorescence in those tubes yet. And that is what we want to see. No time has passed, so we haven't had a chance yet to produce any RNA or proteins. Okay, good confirmation. Nothing is happening when we initially begin. Now we're going to incubate these tubes. We have to incubate at 37 degrees Celsius. So if you have an incubator, use that. That's great. If you don't have an incubator, no worries. You can use body heat for this incubation. So you can hold your tubes in a closed fist. You can put them in your pocket, which is what I'm going to do, and they will they'll heat up and the reactions will proceed from there. Okay. While my samples are incubating, I'm going to go back to my slides. I'm going to explain in more depth what is happening in each of the samples we set up, and then we'll talk through predicting our results. So 4 samples, the first 2 are control samples, 1 positive control and 1 negative control. Tube 1 is our negative control. To that tube, I added only water. So we don't expect any RNA or proteins to be made in that tube. This is just going to show us what nothing looks like in this experimental setup. Tube 2 is our positive control, and there's some great, interesting science happening in tube 2. So I'm going to talk at more depth about tube 2 over the next few slides. We added a special DNA that we engineered called DNA A to tube 2. When we add DNA A to a BioBits pellet, it is going to produce green fluorescent RNA and red fluorescent protein. So when we look back at this tube in about 15 minutes, that's enough time for transcription to have occurred. So we should see green fluorescence in any tube where we have added DNA A. How is it that that can happen? As I told you at the beginning of this presentation, BioBits pellets contain all the components cells use to make proteins, which include the components they use to make RNA. So RNA polymerase, RNA nucleotides. And to illustrate how these components go to work on DNA A, I'm going to use an animation from our Central Dogma Digital Lab. This is part of one of the videos that talk students through the lab, and this animation helps illustrate what the molecules are doing in a way that can kind of make things feel more real to students. So we add DNA A to our BioBits pellet. DNA A contains a special sequence called an aptamer coding sequence. When that aptamer coding sequence is transcribed into mRNA, it folds up onto itself and it binds to a chemical that we've spiked into the BioBits pellet. That interaction between the RNA and that molecule, that chemical, that produces green fluorescence. Aptamers are super cool. They're an engineering trick. They're not really something that happens in nature. We wrote a whole extension on Aptamer Biotechnology if you want to know more about how these work. But one thing that's nice about this lab is that you do not have to go there. That might be TMI for your students if you're teaching middle school, even intro high school. Aptamers are cool, but kind of besides the point to them now. So you can keep it simple for those students. And the takeaway is just green is RNA. Where you see green, that means transcription has occurred, and you can leave it at that. Okay, after transcription comes translation. If we look at tube number 2, in about 4 hours, we should see red fluorescence. How is it that we are able to see red fluorescent protein? Okay, well, BioBits pellets, again, contain the components cells use to make proteins. That includes ribosomes, tRNAs, and amino acids. And so another animation from our digital lab to show what happens next. After we've produced an RNA transcript from DNA A, the end that is not glowing green contains the protein coding sequence. Ribosomes will find that protein coding sequence and they will translate that nucleotide sequence into an amino acid sequence. That amino acid chain folds up onto itself to form a mature red fluorescent protein. This is a protein that naturally produces red fluorescence. And so this part of the lab is a great excuse to dive deep into translation. You can pull out your codon chart. You can talk about translating nucleotide to amino acid sequences. But again, depending on who your students are, that might be TMI. That might be beside the point. So you don't have to go there. You can keep it simple. The takeaway is just red equals protein. Where we see red, we know that translation has occurred. Okay, given what I've told you now about our two control tubes, we should be able to predict our results for just those two tubes. So let's talk through that. Tube 1, again, our negative control, we added only water to this tube. So should we see green fluorescence in a few minutes in tube 1? No, we should not. We did not add DNA to that tube, so no transcription can occur, means no green fluorescence. What about red fluorescence tomorrow? Should we see that in tube 1 when we take another peek at our samples? No, we should not. We didn't add DNA to that tube. No transcription means no translation. So no fluorescence at any time point expected for tube 1. Now, what about tube 2? We added DNA A to tube 2. Again, that's our positive control. And DNA A contains red fluorescent protein coding sequence and then the aptamer sequence that produces green fluorescent RNA. So in a few minutes, should we see green fluorescence in tube number 2? We should. Yeah, all the components are there for that to happen. How about tomorrow, if we look back at our samples in tube 2, should we see red fluorescence? We should, yes. All the components are there for successful translation and production of that mature red fluorescent protein. Okay, this is our control samples. And before I move on from this, I just want to point out two great things about the inclusion of these control samples in our experiment. So one, you can use them to introduce students to good experimental design. What are controls? Why do we use them in an experiment? What do they tell us? So that's a great addition to this lab. The other thing is that simply watching what happens in tube 2, watching transcription lead to translation, is really powerful for students, really good reinforcement of everything you've told them about the central dogma. So with tube 2, they don't have to just take your word for it. They can see what's happening in real time. They can see the central dogma play out in their own samples. So that's kind of one advantage of using this lab, is to kind of, you don't have, seeing is believing, right? Students actually see the central dogma happening in real time. But another layer of power in this lab comes from tubes 3 and 4. These are our two experimental samples. They have been manipulated in some way to interfere with gene expression. And so by investigating what's happening in these tubes, students get to walk through some critical thinking. They get to do some inquiry here. Their job will be to explain how gene expression is being derailed. So is it that transcription was affected, or was translation? And was the failure due to faulty cellular machinery? Did something about the components that cells use to read DNA break? Or did the DNA message itself change? In tube 3 is like a twist on tube 2. We added DNA to this tube, to tube number 3, just like we added to tube number 2. DNA A contains the aptamer sequence, the red fluorescent protein coding sequence. But we also spiked in another chemical, something called kanamycin. So students' job is to determine the effect that kanamycin has on gene expression. Tube number 4 is our second experimental sample. To that tube, we added DNA B. DNA B is a mutated form of DNA A. And students' job here is to determine what effect that mutation has on the instructions that were written into DNA A. Okay, at this point, our samples have been incubating for not quite 15 minutes, but perhaps enough time for us to see a result. So let's do our first observation. So grabbing my samples from my pocket, here they are. We are putting those in our P51. I think my orientation is right. Yes, okay. Turning on the light. Let's see what we see. Okay. There's some droplets there, but we see bright green fluorescence in 3 of our tubes. And with that view, it might even be hard for you to tell which 3 tubes we're looking at. So I'm going to switch to my slides. my results that I'm seeing here match the photo of expected results on this slide. So we see no fluorescence in tube number 1, but bright green fluorescence in tubes 2, 3, and 4. So what does that mean? Again, green is our indicator of transcription. So no transcription in tube 1 as expected. We added only water to that tube, but successful transcription in tubes 2, 3, and 4. Great, okay. What about day 2? Our second time point is when we, or I guess technically our third time point is when we assess translation. Now, cooking show style, I have some tubes that I set up yesterday so you can get a sense today of what our samples should look like. So I'm putting those in my P51 now. Turn my slides off for a moment. And I'm going to show you what my day 2 samples look like. Okay, so here you should see bright red fluorescence in tube 2. So the tube at this end is tube number 1. The second tube over, tube 2, there's where you're seeing some red fluorescence. Now in tubes 3 and 4, you might be seeing some residual green fluorescence from our RNA, but certainly not seeing any red fluorescence. I'm going to switch back to my slides where I have a photo of the expected results that matches what I'm seeing in my P51. And that confirms no red fluorescence in tubes 3 and 4. So what does that mean again? Well, that means translation failed in tubes 3 and 4. We had successful transcription on day 1, but then on day 2, no translation. So let's talk more about what might be happening in each of those tubes. So in general, there are 2 ways to interfere with protein expression. One possibility is that the DNA message has been disrupted. So if we garble the DNA message, we put a typo in the DNA message, we will impact the resulting protein. Alternatively, it might be that the message stays the same. The message is just fine, but the machinery that cells are using to read that message has been broken in some way. So I wonder if we can determine which of these possibilities is at play in tubes 3 and 4. So let's focus first just on tube 3. So in this table, I'm comparing tube 3 to tube 2, just to simplify things. So tube 3, we added DNA A, the same DNA that we added to tube 2, but then we also spiked in another molecule called kanamycin. Now, because the DNA that we added to each of these tubes is the same, that should indicate that the DNA message was not the culprit here. We know DNA A is working fine because we're seeing the expected results in tube 2. So that must mean that we have disrupted the machinery that cells use to read DNA. When I mentioned kanamycin, you might have noticed that that sounds like the name of an antibiotic, and that's because it is an antibiotic, and it works by inhibiting bacterial ribosomes. So we produced an RNA transcript in tube 3, but that transcript could not be translated into an amino acid chain because our ribosomes had been inhibited by kanamycin. Okay, what about tube 4? This table, again, we're comparing tube 4 to tube 2 for simplicity. To tube 4, remember, we added DNA B, which is a mutated form of DNA A. So that should give us an indication right away that we have disrupted the DNA message. There was no other agent at play. We changed the instructions that were written into DNA A to make DNA B, and that is what was happening in this tube. Now, what type of mutation would have caused this to happen? Well, that's a great question to ask your students. If you've taught them anything about mutations, they should be able to generate ideas about what type of mutation might have led to the observations that we made here. I'll tell you for your own information that we basically deleted the entire protein coding sequence to turn DNA A into DNA B. But you don't have to tell your students that. They can generate their own ideas. Perhaps a premature stop codon terminated translation of the red fluorescent protein too early. Perhaps there was a deletion that took a big bite out of the protein coding sequence. This is a great opportunity for them to kind of use some creative and critical thinking to apply what they already know about DNA to their observations in this experiment. So that's our Central Dogma Lab. Hopefully you've seen this as a great way to augment your core curriculum by adding a layer of hands-on experimentation. We watched transcription and translation as they were happening in real time, And then we also explored different ways of interfering with gene expression and used our observations to infer what's happening on the molecular level. At this point, I now want to talk about classroom implementation. What would it take to bring this lab to your students? In terms of materials, you'd need a Central Dogma Learning Lab kit. That includes reagents for 8 student groups. In terms of hardware, you need a blue light visualizer. You need some way to visualize the fluorescence in your samples. Today I used a P51 viewer. That's one option, but it's not the only option. Any blue light visualizer would work here. So if you happen to have the Blue Gel by Mini PCR, that's our electrophoresis system, you can visualize your Central Dogma Lab samples in the Blue Gel. You also need a micropipette that can dispense 4 microliters. Today, again, I used a fixed volume mini pet, but you can also use a variable micropipette as long as 4 microliters is within its range. In terms of time, it takes about 15 minutes of teacher prep to get ready for this experiment. That's just portioning out your reagents and preparing your BioBits pellet strips. In class, it takes about 30 minutes to do the parts of the experiment that I demonstrated for you today. You set up your reactions and you observe transcription. The next day, you would observe translation, and that's a very brief amount of time, like 5 or 10 minutes, just to turn on the P51, take your observation, and debrief what you're seeing. The timing of day 1 relative to day 2 can be flexible. So I think most folks do it next day, but if you're like on a rotating block schedule and maybe you don't see your students every day, you can come back 2-3 days later, it's fine. Once you've produced that red fluorescent protein, it's pretty stable. So even like a week after setting up your samples, they should look more or less the same as they would have looked on the next day. The curriculum for this lab, as I mentioned, is free, so you can go to our website, download the teachers and students guides. This is one sample of one page from our students guide. This is a visual lab guide, so what we do is we try to convey as much information as possible through diagrams with brief bulleted captions. There are assessment questions interspersed throughout, and those are also available as an e-worksheet that you can integrate with your LMS. There are optional extension activities that add on to the core lab that are focused on reviewing transcription and translation, focused on gene expression, and on Aptamer Biotechnology. And these extensions are really what make this new curriculum so powerful. This basically becomes a modular lab, so you can take the core lab and then augment it with whatever extensions are right for your students to tailor this to the level at which you teach. So this lab, again, is truly suitable for students in middle school, high school, or college and beyond. And so I'll speak over the next slides to some sample implementations, how you might implement this curriculum for your students depending on which level they sit at. For middle school, we would recommend just completing the core Central Dogma Lab. No extensions included. The core Central Dogma Lab will be enough at that level. And you can focus your discussion around the idea that expression of a trait can be disrupted by a DNA mutation. So in this case, our trait is red fluorescence. And in tube 4, as we saw, that expression of that red fluorescence was disrupted by mutating our DNA. If you focus the discussion in that way, it would service NGSS Middle School Standard LS3 very well. For high schoolers, we would recommend completing the core lab and then adding on the extension on reviewing transcription and translation. In that extension, it really goes deep into the molecules that coordinate transcription and translation, what their interactions and roles look like. So it provides a little bit more depth to our core lab. If you implemented the curriculum in this way, you would service NGSS High School Standards LS1 and LS3 by focusing the discussion around visualizing the DNA to protein process with transcription and translation as the intermediaries. If you're teaching AP biology, advanced biology, college level biology, you would pick and choose the extensions that are needed to cover your specific learning objectives and standards. So you'd complete the core lab and then you would mix and match the extensions depending on exactly where you need coverage. On the slide here, we've taken, for example, the AP Bio topics. So our review of transcription and translation extension covers topics 6.3 and 6.4. Our gene regulation extension really goes deep into the different parts of a gene. It can cover topics like promoters, and that covers topic AP Biotopic 6.5. We also have an extension on the Aptamer Biotechnology. I mentioned this earlier. It goes really deep into how aptamers work and all the different scientific applications of aptamers. And that would cover the biotechnology-oriented AP Biotopic 6.8. Now again, AP Bio is just shown here for example. The key would be tailoring your use of extensions to your curriculum. But by mixing and matching those, you can reach students at really any level who are learning biology. You can download all of those extensions, all of our curriculum from our website. If you go to the Central Dogma Lab page, click the Curriculum Downloads tab, and you'll be able to download the Instructor's Guide, Student's Guide, slides, and more. As I mentioned, we also offer the Central Dogma Digital Lab, which is a virtual version of the Central Dogma Lab, and this can be used with or without the hands-on kit. This digital lab really supports deep student engagement with this material, and you can use it in a couple different ways. So this digital lab includes two versions. One we call our Lab Companion. This is basically an interactive lab guide. You use it to guide the hands-on lab. Each step of the protocol is illustrated with photos and videos, so students really have a good sense of what their sample should look like each step of the way. We also offer a lab simulation. This is a fully virtual learning experience that can be completed in about 30 or 45 minutes. It is great to prepare for the hands-on lab, or it could replace it entirely. No matter which version you implement, you have access to teacher tools that are great for progress monitoring. Those include our performance snapshot, which goes through the lab question by question, showing how many of your students got each question correct, incorrect, or correct in multiple tries. A great tool for real-time intervention. So if you see that half of your students are getting a question wrong, you can stop them and correct whatever misconception is causing that to occur before it has a chance to take root. All of our digital labs include narrated videos hosted by a friendly scientist that bring experiments to life and walk students through what is happening molecularly at each step of our experiment. There are auto-graded assessment questions on virtually every screen, so students get immediate feedback constantly about their understanding. And all of our digital labs include authentic lab results. So even if students who are doing the hands-on lab make an error, don't get the expected results, they can still refer to these videos and photos of the expected results and walk through interpretation. Digital labs can offer you a little bit more flexibility for how you're implementing the Central Dogma Lab. So for instance, you can use the Lab Companion to guide the hands-on lab in lieu of a printed lab manual. You can also assign the lab simulation as pre-work to make sure your students walk into your classroom prepared. The lab simulation is actually probably the best homework assignment you could give a student who's about to embark on the Central Dogma Lab. They will walk through every step of the experiment. They'll get a really good conceptual understanding of this lab, and when they come to your classroom the next day, they will be really well prepared to focus just on the hands-on mechanics of completing the experiment. You can also use the lab simulation as a makeup lab in the event of classroom absence. So if you have an absent student, instead of trying to schedule time for them to come in and make up the hands-on lab, you can assign the lab simulation. They can complete it on their own time, and it's a bit more flexible for you and for them. If you're going to be absent from the classroom as well on a day when you have a hands-on lab planned, you can also switch and implement the lab simulation instead of the hands-on lab. That's certainly easier often than giving a sub plan that involves doing a hands-on experiment. So that's our BioBits Central Dogma Lab. I hope you enjoyed our demonstration today. Again, this lab is a great way that you can add hands-on experimentation to your coverage of this essential curriculum area. We watched along as RNA and proteins were produced in real time. We also looked at different ways gene expression can be disrupted. Thank you so much for joining me. I hope you found something today that you want to bring back to your classroom.